The subject botany in the first semester, particularly in the University of Mysore syllabus. Session number 11. Is Introduction and General Classification of Bacteria and Archaebacteria. Introducing the topic, how to define bacteria. Bacteria are small, microscopic, unicellular, prokaryotic microorganism. So therefore, the study of bacteria is called bacteriology. Historically, the credit of discovery of bacteria goes to Anton van Leeuwenhoek in the year 1976. He named as an animalcule. It was renamed as bacteria by Louis Pasteur. Therefore, Pasteur is oftenly referred as father of bacteriology. So bacteria are omnipresent. They are present in air, water, soil, milk, temperate, Arctic, tropical regions, in the hot springs, in the plants and animals, and even in human beings. And it can withstand extreme temperature of 190 degrees centigrade. On an average, the cells of bacteria ranges from 1 micron to zero point 0.1 micron to 1 micron, and some forms may as large as 15 micron. Coming to the classification of bacteria. Bacteria are classified on different criteria based on the cell shape. 4 morphological forms have been identified in bacteria. They are Bacillus, Caucus, Spirillum, and comma shaped. Let us see one by one. Bacillus. In this group, the cells are rod shaped and elongated. They are second largest among bacteria. Example, Mycobacterium. Second is the Caucus. In this case, the cells are spherical. These are smallest among bacteria. Example, Streptococcus, Staphylococcus, etc. Third group is Spirillum. The cells in this group will be in the form of loose spherical, and these forms are largest among bacteria. Example, Rhodospirillum. And the fourth group is comma, that is comma shaped. The cell will be in the form of punctuation mark. Example, Vibrio. Therefore, the Caucus forms exist in single cell, they are called as Micrococcus. In double or in pairs, they are called as Diplococcus. In chain, called as Streptococcus. Sometimes they are found in arranged in the form of sheet, namely Staphylococcus. And many bacteria exhibit several shapes, and this is known as pleomorphic, and the phenomenon is known as pleomorphism. Example, Rhizobium. Except Caucus, other forms possesses organs of locomotion, that is called flagella. Flagella is long thread-like structure whose length is usually exceeds that of the cell. It arises from basal granule. On the other hand, the other classification of bacteria is the bacteria are classified again on the number and position of flagella into 5 types, that is Atrichus. In this case, flagella is absent, no flagella. Example, Caucus. Monotrichous. In case of monotrichous group, there will be a single flagella present at one end. Example, Vibrio. Third group is Lophotrichous. In this case, many flagella present at one pole. Example, Spirillum. Fourth group is Amphitrichous. In this group, two tufts of flagella attached at either pole of the cell. Example, Spirilla. And the last one, that is Peritrichous, is the group in which the flagellas are present all around the cell. Example, Salmonella typhimurium. Not only these two types of classification, that is based on the shape of the cell and the number of flagella, the bacteria are also classified based on staining methods. That is called Gram staining. This was for the first time Hans Christian Gram in the year 1884. He classified bacteria based on the staining technique, that is into gram positive and gram negative bacteria. That's about bacteria and their classification. Coming to another group called as Archaebacteria. Archaebacteria are again a group of bacteria. They are found in harsh environment and they are found in undersea volcanic vents and acid hot springs, or they are also found in salty water, hence they are called saltwater Archaebacteria. Therefore, these Archaebacteria lack peptidoglycan in the cell wall. Normally, the bacteria have the peptidoglycan in the cell wall, whereas in this group, Archaebacteria, peptidoglycan is absent. And they have different lipids in their cell membrane. And ribosomes are different. And the gene sequence in this, particularly in this group of bacteria, that is Archaebacteria, is completely different compared to the normal bacteria. Therefore, they do not require oxygen and can live in extremely salty environment as well as extremely hot environment. Therefore, Archaebacteria can live in extremely harsh environment and they are called as the ancient bacteria. And they are divided into three subgroups, that is Methanogens, thermoacidophils and extreme halophiles. Let us see one by one. Methanogens. Methanogens are a group of Archaebacteria in which they live in anaerobic environment. There will be no oxygen and they get energy by changing hydrogen and carbon dioxide into methane gas. They are found in swamps, sewage treatment plant, digestive tracts of animals. Second group is thermoacidophils. These are also called thermophiles. They live in extremely hot environment and they are found in volcanic vents, hot springs, cracks on ocean floor that leak acids. And the third group of Archaebacteria are extreme halophiles. These extreme halophiles, they live in very salty water. They use salt to generate ATP as energy. And They are found in Dead Sea, Great Salt Lake are inhabitants of these groups of bacteria, that is extreme halophiles. Later we will see what is the importance of these bacteria. So bacteria are economically important as they are used by human for many purposes. The beneficial use includes production of traditional food such as yogurt, cheese, and vinegar. They are also important in agriculture for the compost and fertilizer production. They are used in medicine for the production of antibiotics, insulin, and so on. They are used in medicine for the production of antibiotics, insulin, enzymes, and lot of other medicines, so on. And also, they are helpful to degrade the dead animals and plants and bring valuable nutrients back to the earth. Some species also clean harmful pollutants of environment by bioremediation. So therefore, bacteria are in human life a harmonious existence with human cells. But diseases and infections can be caused when this balance is disrupted, immune system is weakened, the bacteria can transfer genetic information between one another, and this is one of the means by which antibiotic resistance is spread amongst the bacterial population.